Hello everyone, welcome to Biochemistry 1 for the Spring 2023 term. So my name is VJ Antharam, and I thought I'd take this opportunity to go over some of the requirements and prerequisites for the course. So first of all, this is Biochem 1, Biology 430. We're going to meet on the campus of Fort Bragg, and our scheduled times are Monday and Wednesday, 6 to 10 p.m. And the room is going to be at JSOM. The actual room number is yet to be determined, so we'll probably just meet somewhere there and decide upon a room. Class is going to meet March 13th, is my understanding, and this is the first time since 2020 that I've taught in this class, the 8-week class. It's going to be very compressed, so let's see how far we can go in learning this material. So a brief description about myself. I got my bachelor's and PhD from the University of Florida. I also did 2 postdocs from there, so mostly my research involved some biophysics and some biochemistry, and I spent a very long time in Gainesville, and it was a very fun place. Also, there was a lot of stress as well, and I was there for almost 15 to 16 years. I got this job here at Fort Bragg as an adjunct at around 2018 or so, 2018. So it's been about 4 or 5 years since I've been at Campbell. So a little bit about the way this course is structured. Once again, it's 8 weeks, so it's going to be very fast and very compressed. And in the midst of all of that, we'll have to try to squeeze in some labs as well. I divide this course into 4 modules. The first module is protein structure and function, just learning about the 3-dimensional structure of proteins. The second modules are looking at carbohydrates, sugars such as glucose and fructose, as well as lipids. So lipids are a very large class of molecules. It's not just the membrane. So usually I combine these 2 macromolecules, important molecules in the cell, as one unit. So this would be our second module. Our third module is looking at enzymes and studying enzymes. And then the fourth module, the final module, is looking at basic metabolism, some glycolysis, maybe the TCA cycle. There's so many different metabolic pathways. You can see how much we can fit in this module. Metabolism is probably one of my favorite topics, as well as protein structure and function. But we will not go too deep into metabolism as much as I would like to, but that's okay. Most of this course is PowerPoint based, so if you had me in a previous class, such as general chemistry, I write a lot, but here I'm going to talk a lot, and you'll see a lot of cartoons, you'll see a lot of visuals, and you'll see a lot of slides. So most of my lectures are going to be on PowerPoints, and most all of the PowerPoints should already be on Blackboard. I also have some videos, not as much as in Gen Chem, because there's really no problem set. This is, very more information dense material. All right, let's talk a little bit about grading here. About 10 drills or homework assignments worth 15% of your grade, and those are easy 10 points. Each one is 10 points, so take advantage of that. Many times I'll ask you to go outside of the textbook to talk about areas of biochemistry that are hopefully related to the real world. There'll be 2 exams. The first exam is on Module 1, which is protein structure function. The second exam is on Module 2, carbohydrates and lipids. And the final exam, which is scheduled for the last day of class, will cover both, compress them both, enzymes and metabolism. I'm going to try to squeeze in some labs here, hopefully. It's depending on if I can get the materials ordered and if we have space and enough people. Mostly enough chemicals and materials. The first lab is just practicing micropipetting. The second lab is using a pH meter. So hopefully JSOM will have some pH meters. I'll talk to the TAs about that. We have two labs that are you can do at home. One of them looking at the internet, informatics. Another one is downloading the software called PyMol. So you can check out Blackboard on how to download PyMol. And then maybe another wet lab about studying how to diagnose lipids or maybe how to diagnose peptides. I still really haven't decided what lab we can do for this final lab. And so that is around 15%. I'd like for you to keep a lab notebook. So with a lab notebook, you can write out your lab, and you want to write it like an NIH style lab notebook. National Institute of Health is what the NIH stands for, and I'll give you the basis for that. This is not something you need to be stressed out about, but a certain way we want you to write the lab notebook, and I'll give you that little sheet that you can use as a guide. Once again, everybody's lab notebook is going to be different. The best lab notebook is one that you can teach off of. Here's a note on the textbook. I believe there's a new edition available of this. This is the one that I've been using for the last 5 or 6 years. And I guess it's enough for this beginning biochemistry course. So if you're looking to save some money, you can get this. You can rent it. I would recommend purchasing it. It's not required. It's not mandatory. It's just highly recommended. So if you're on a financial pinch, you can use my PowerPoint notes and the associated videos to get yourself a good grade. And this is the grade scale. 90 to 100 is a B. Sorry, 90 to 100 is an A. 80 to 90 is a B, so on and so forth. Actually, if you're at a 90, I'll bump you up to an A. I think I will end this because I'm probably going a little bit too long for my liking. And there's just one assignment I'd like for you to do. I'd like for you to make some flashcards of the 20 amino acids. On one side, you'll have both the 3-letter amino acid abbreviation and the 1-letter amino acid abbreviation. If you're using the Voigt and Voigt textbook that was on the previous slide, I believe they're on page 68 to 69. And on the other side, you want the structure of the amino acids. Now, I wanted you to write it in a very specific way. Now, your textbook, if you use Voigt and Voigt, has it at pH 7.2, which is not how I want you to write and do this assignment. I want you to write it totally protonated and have all the pKa's labeled. So write the most protonated form and write the pKa's labeled. If this is confusing, you can watch a 4-minute video that's on Blackboard that talks a little bit about that in detail. Well, here's an example of this. So on one side of the flashcard, alanine, the 3-letter amino acid abbreviation is ALA. A is its 1-letter amino acid abbreviation. And here's the structure that you would draw of alanine. So most amino acids will have the alpha carbon, the side chain, the alpha carboxylic acid. You will want to label that H along with the pKa. Okay. Your textbook will not have that H if you're using Voigt and Voigt because at pH 7.2 physiologic pH, it's gone. It's deprotonated. And then you're going to write the pKa of the alpha amino proton. So at pH of 7.2, the proton is on, so usually your textbook will have this fully protonated. So it's a little different. I don't want you to write it as is the textbook. I want you to write it totally and fully protonated. So we'll have more information on that 4-minute video if you need more clarification. You can also come see me during class. So I will end this welcome video and have a wonderful day. Let's have a good class. It's going to be kind of rushed and for 8 weeks. And I hope you like the course and you'll enjoy the course. So thank you, everyone. I'll talk to you soon. Bye-bye.